you.
Thank you very much for the introduction. And it's really a big, a very great honor to be here together with so many famous experts in this field. My topic today is about protein-based pneumococcal vaccines. Currently there are many pneumococcal vaccines being approved in the market. All is based on the polysaccharide. Some of them are conjugate vaccine from PCV7 to 13 to 15, 20, and now there are 24 and 31 valent vaccine in the pipeline. So it becomes more and more prevalent. And all we know, when the valent becomes more and more, it becomes more complicated and difficult in terms of CMC part. And also there are some interference of the immune response because of the heavy burden of the carrier protein. So actually there are some other ways to approach the cross-serotype protection against the different pneumococcal serotypes. One of them is protein-based vaccine. So this is my topic today. Because I'm from clinical department, there are 3 points today, so I mainly focus on the last part, the clinical study. And for the previous two, maybe I go through them fast. So CanSino's protein-based vaccine, pneumococcal vaccine, is use PSPA protein as our antigen. We all know that there are several proteins in pneumococcal can have the potential to become a vaccine antigen, including PSPA, PSAA, and Ph.D, some other proteins like this. CanSino use PSPA. And PSPA surface protein in pneumococcus, and it can prevent, they have many functionals to prevent this bacteria to be killed by a human host immunity system. When we use PSP as the antigen in a vaccine, there are always two major concerns about this PSPA. One concern is the diversity of PSPA. And we know there are 3 family, 6 clade of this protein. So there are different variants of this PSP protein. So as a vaccine antigen, we need to have a good cross protection. And another concern is the safety concern from some kind of PSPA variants has the homology with myosin. So we do have the strategy to address these 2 concerns. So for the diversity, we use 3-valent PSPA, just as you say here, P3296 and P5668 and PRX1. These 3 are variants. It can provide over 98% prevalent strains in China. And another thing is we do some modification to PSPA protein to avoid its cardiotoxicity potential risks like this. And this was actually from Dr. Zhang Jinren's team. Maybe he has already introduced some details of this liver capture model the day before yesterday in his presentation. So I do not want to go deeper in this model. Currently, we cooperate with Tsinghua Dr. Zhang Jinren's team. We try to use our clinical serum sample to use in this liver capture model to show some protection results. In the right side, you can see this is all already been done in a mice model. We can see some bactericidal activities for the for PBPV, that is protein-based pneumonia vaccine, we short as PBPV. For PBPV antibodies, in a passive protection model, we already see some effect. You can see we can reduce the bacterial load in the blood and most of them go into the liver, being captured in the liver, then being killed. And we have sent our phase 1 trial serum to his lab, to Dr. Jones' lab, and I think testing it on our way. Maybe several months later, we can see some results of this. Second part is the preclinical study, and actually we have done a lot of them. I just put 3 slides here. First one is in mice model, and it's an efficacy evaluation. You can see we use different dosage of this candidate vaccine. It seems it all works very well. It gave the fully protection in this animal model, and control group we can see many deaths, and for the vaccine group, all 3 doses group, all the animals are survived. And this is immunogenicity data in the NHP model, and we can see the good immune response. This is serum IgG level. Post-vaccination, we have a strong response. I don't want to present more about this because later on we can see some clinical data. And this is another slide for the animal model. We use another assay, that is modified MOPA assay, to evaluate the functional antibody level against pneumococcus. So here we go. We come into the clinical study. Till now, we have finished 2 clinical trials for this candidate vaccine. First one is first-in-human study in adults aged 18 to 49 years old. We conducted a trial in China in 2020. And this is, I think, the total sample size of this trial is 120. And we use randomized double-blind and use placebo control. This is the dose escalation study. We have 3 dosage: low dose, middle dose, and high dose, from 20 microgram per dose per antigen to 100 microgram per antigen per dose. And also we have 3 dose schedule in this trial, and take some blood samples in different time points to evaluate the safety, laboratory safety indicators, as well as to see the antibody connects. Sorry. Okay, here is the safety profile. It's a preliminary safety profile from the first-in-human trial. This slide shows you the adverse reactions within 30 days post-vaccination, each dose. Generally speaking, we do not see any special risks post-vaccination within 30 days, especially those adverse reactions related to the cardiotoxicity and hemolytic toxicity. So that because I mentioned the risks previously, so these 2 kinds of AR is listed as the AESI, that is adverse events of special interest. We do not observe this kind of risks during the process. And it seems all the dose group were overall tolerated well. And more ARs in the high dose group, as can be understood, and the more local site adverse reaction observed as the dose and the dose increased. So, but most of them are mild and transient, and we do not observe any grade 3 level adverse reactions in this trial. And this is the immunogenicity data again, from the IgG antibody. I think this is very interesting data here. You can see the dynamics of the antibody. It's different. It's different from PSPA protein antigen and the PRY protein. Yeah, PRY is another antigen in this vaccine. it seems for PSPA antibody, it peaked after the first dose, and subsequent doses did not, induce a higher antibody level, and it seems there is a dose-response relationship, but the difference is not that large, especially for the low dose and middle dose. So we can see for the PSPA, this antigen, it shows a pattern of booster immunity in this age group. And PLY protein is a little difference. And we can see the antibody level peaked after second dose. And the third dose do not induce even higher antibody level. And it shows a significant dose-response relationship. And so we can see adults in this age group, PLY, this antigen shows a primary immunity pattern. So I think this results does not surprise us because PSPA is a surface protein, and pneumolizing, the PLY, pneumolizing is protein inside the pneumococcus bacteria. So it shows this pattern. And this is MOPA results. MOPA gives us some information of the functional antibody level. And we use different strains from different PSPA families, from family 1 clade 2, family 2 clade 3, and family 2 clade 4. Generally speaking, I think the OPA bacteria, the OPA antibody level is generally similar trending as IgG results. We do not see, difference between the different dosage, and no great difference between different, schedules also, except for one strain that is TRAP6A, because for this strain we use a different, method, different assay. MOPA assay has always been used to evaluate the functional antibody level for the polysaccharide-based vaccine. So we do some modification to OPA to make it work for this for this PBPV vaccine. It shows some activities in this assay, but I need to say maybe this assay is not as suitable for a protein-based vaccine. We can just use it to see some trending, but I don't think this is best appropriate assay for this kind of vaccine. And then, based on the result we got from first-in-human trial, we went into phase 1B trial, 2023. The target population is elderly, 50 years old and above. So in this trial, we just select the middle dose. So after analyze, we think the middle dose is our target dosage. And then we use just a single dose schedule for the vaccination. And in this trial, we do not use placebo control. We use a positive control. That's a polysaccharide vaccine, PPS with 23 from MSD. And the safety profile, I think it is similar to the first-in-human trial, but you can see the adverse reaction observed in this trial is much more less than the first trial. That can be understood because this usually when we conduct a trial in the elderly people, the adverse reaction will become less than in the younger adults. So from this trial, we got a message. The PBPV vaccine, when we use this kind of vaccine in the elderly people, the safety profile is okay. And this is the antibody response elicited by PBPV and versus PPSV23. This is IgG antibody level. Because this is the IgG against the protein antigen, so you can see for the control group, there's no response. But for the protein group, vaccine group, we can see a really strong response and relatively good immune persistency within 6 months. And this is for another antigen. The interesting thing is for pneumolizing antigen, it seems the pattern is not that same as the PSPA protein, especially for the immune persistency. It seems that for pneumolizing antibody, it drops faster than the PSPA. So as we say, because this shows the primary immune pattern, not a booster. Always within a booster vaccine, post-booster vaccine, the immune persistency is better than the primary one. So it makes sense. And also, this is the MOPA results. We still use some strains as the target strain. For the family 1 clade 2 strains, these 2 strains is not included in the control vaccine. PPSV23 not include serotype 6A and F38. So we do not see any response, MOPA response against these 2 serotypes. But for other 4 strains, we see a different pattern. In 2 strains from family 2 clade 3, the PBPV antibody level is higher than the polysaccharide vaccine. But for another 2 from family 2 clade 4 strain, and the polysaccharide vaccine has the higher antibody level than the protein vaccine. I think that is because, just mentioned before, that is because the protein vaccine if the protein vaccine want to get function against the bacteria, the antibody need to be reached this protein itself. But all we know, there is a polysaccharide capsule around this bacteria. So sometimes when this capsule become thin, then this protein can reach this antibody can reach to that protein. So that is why we see some different results. That is also why I said maybe this MOPA is not a very good assay to evaluate this protein-based antibody. But now we have we are developing other assays, some assays just like Dr. Zhang Jinren's team. Maybe they can give us better answer. Okay, here is just a very simple summary. So PBPV is, till now we have 2 trials, and we see a good safety profile till now. No other special safety concerns. And also we see PBPV has good immunogenicity response in human body. And PSPA shows a booster pattern, and pneumolizing protein shows primary immunity pattern. And we see it works till now. And next step, what we want to do is to start proof concept phase 2 to preliminary evaluate this vaccine's effectiveness in human. Okay, thank you.
Thank you very much. Any questions?
Dr. Gou, I was wondering, is this subunit vaccine much cheaper than the PPSV 23 even?
Yes, of course, it is a subunit, much, much cheaper than the current conjugate vaccine, and much easier to produce and have a good lot-to-lot consistency. Yeah, I think that's a big advantage.
What do you project the cost-wise? How does it compare with PPSV?
PPSV, I think, I'm from clinical team, so I just give you a roughly estimation. I think that is 1/10, yeah, compared to the PPSV. That is a polysaccharide, not conjugate. If we compare to the conjugate vaccine, more than that. Yeah.
Do you think mixing the protein with the capsule, which is still cost-effective, would that promote the distribution of this vaccine or the efficacy?
For this vaccine, I think definitely we need an efficacy study to prove its effectiveness in population. So this is just a preliminary immunogenicity study. So later, after the POC phase 2, we'd like to maybe cooperate with somebody to start an efficacy study. Thank you.
I'm sorry. I was curious that you chose control to use the pneumococcal, the polysaccharide vaccine, and not the conjugate vaccine. What was the rationale? Because I would have thought that
Yeah.
So compare protein to polysaccharides, not that fair.
Actually, the reason is very simple, because in China, there's no conjugate vaccine can be used in adults till now. Yeah, so we just use polysaccharide.
Fair enough.
Yeah.
Okay, any other question? If not, thank